Welcome to Decoding Healthcare Research. I'm your host, Efrain Riveros, Dr. E.F. Rain. Today we will be talking about alcohol, also the animal models to study alcohol, and in particular, we are going to talk about tolerance and some relationship with the circadian cycles. We came across a very important paper, and we have the author of this paper. We have Dr. Anthony Lange. He's a postdoctoral research scholar of the Department of Neurosciences and Pharmacology at the University of Iowa. Welcome, Dr. Lange.
Thank you.
Thank you so much. Thank you so much. So we will be discussing his paper, Alcohol Sensitivity and Tolerance Encoding in Sleep Regulatory Circadian Neurons in Drosophila. This was published in 2023 in the journal Addict Biology. So the insights that Dr. Lange is going to give us are very enlightening in understanding the different associations between alcohol and tolerance and what would be the implications for human research. So first of all, I'm going to ask you, Dr. Lange, what sparked your interest in this topic?
So a couple of things. A primary interest is just in neuroplasticity, of which alcohol is kind of the ultimate tool for neuroplasticity in the brain, because it's kind of unique amongst drugs in its level of we'll call it promiscuity. In essence, you can, like, if you name a receptor in the brain, there's a decent chance alcohol does something to it, which is weird. Usually a drug interacts with one or two different types of receptors, right? So like nicotine acts with nicotinic receptors. That's why we named them as such, right? Cocaine interacts with dopamine, right? It interacts with that system. Alcohol is going to basically do everything. It's a very small molecule, so it can just kind of get around and cross the blood-brain barrier very easily and just interact with a whole bunch of different molecules. And then the reason to look at tolerance is that this is the first form of plasticity to really take place. So tolerance, we in the research field, at least, we divide it into three different forms. You have acute tolerance, which is more at a cellular level, and it occurs like while you're having that first drink. Then you have rapid tolerance, which is what myself and my mentor, Dr. Wolf, were interested in, which is the tolerance that develops after you've metabolized that first drink. Right. So if you go out, you have a drink, the next night, the alcohol will affect you a bit less. Tolerance, which my old lab has been looking into, and they do have some stuff out there on that as well, looking at Drosophila, and that's what most people think of probably when they think of tolerance. So it builds up over time, over many repeated exposures that are not always intoxicating. You can just have a single drink a couple of times a week, you'll build up a tolerance, even though you're not getting intoxicated. Rapid tolerance is usually results from a intoxicating dose, at least the way we study it. And we do feel that's relevant from like a healthcare standpoint, because at least in kind of American culture, related to like Northwestern European cultures, binge drinking is kind of the most common form of consumption, especially for a first exposure, right? People are drinking for the first time in high school or at college, right? A lot of times, and it's at a party situation, so they're going to have a decent amount of drinks. Mm-hmm. So we want to figure out, okay, so what's this doing to the brain? And the easiest, and the first form of plasticity that often takes place is tolerance. And we've known for right? Decades of tolerance is correlated with risk for alcohol use disorders. People who have a higher innate tolerance are more at risk for developing alcohol use disorders, and it tends to be more severe in them, just because you have to consume more to get the same effects.
Yeah, and the background is fascinating, especially because of
Mhm.
this association with alcohol use disorders. Is not only the acute effect, but how it's going to affect behavioral changes over time. And as I mentioned to you offline, it's not usual that we discuss animal research in this podcast, but this paper in particular definitely caught our eye because we
Mhm.
think it's important to understand the parallels between the models that you guys use and actually
Mhm.
the human brain. So in that regard, you use Drosophila. So what is Drosophila and why is this a valid model?
So Drosophila, it's the fruit fly. It's one of the gold standards of um, this type of preclinical work, along with the mouse. Those are kind of the two really big gold standards for looking for interactions of any sort of genetic type manipulation and behavior. And so Drosophila is very useful for this work because they respond to alcohol actually in the same way humans do. So one, they develop kind of all the big behavioral responses. They develop tolerance, they develop a preference for alcohol, they develop a positive association with it over time. Initially, they find it aversive, but the next day they'll find it a positive, a reward. So they'll get a reward out of it. And they do seem to develop a withdrawal as well. Which it's a little difficult to test withdrawal in fly, but basically when you expose them to alcohol and then take it away, they become more susceptible to seizures, which is associated with withdrawal. Additionally, their behavior just over the course of the sedation, we're exposing them to alcohol vapor because you can't really get a fly to drink on command. But as you expose them to the alcohol vapor, they actually enter a state of hyperactivity. So they'll start moving around more quickly, in the exact same way that people do, right? That's one of the reasons we're using it for, is that social lubricant. You get, it has a kind of an upper effect at first before it becomes a depressant. So they respond to it in all the same ways. And then at the neural level, they tend to use the same, or at least very similar neurotransmitters. In the case of this paper, it's the same type of neurotransmitter that we found. It's glutamate, very common excitatory signaling molecule. And a lot of their systems are very similar, even though we're separated by several 100 million years of evolution. They still have circadian rhythms. They still respond to light and morning. They're, and they're a gregarious animal. They're social. So even with that, sticking with the alcohol theme for this, if a fly experiences social rejection in some way, or it's isolated socially, they will consume more alcohol than
I
a fly
see.
that hasn't had these experiences. So it affects them in very similar ways. They do need to seek it out a little bit because they are fruit flies, so they are looking for rotting fruit in some manner to lay their eggs. So they have a little bit of an innate kind of system in there to sense it as well.
I see, and I am sure that the experimental setup has a lot of complexities. But can
Mhm.
you walk us through the methods in an
Yeah,
easy to understand way?
yeah. So ultimately, we're basically performing an kind of equivalent to a field sobriety test. So once again, in line with they respond to alcohol in very similar ways. They will lose coordination of behavior, which is exactly what the police are looking for in a field sobriety test. We take it a little further than what would happen there. We are looking for what's called basically kind of the loss of righting reflex. So we have flies usually around 20 flies in a test tube, and 8 test tubes then across a kind of rack that is loose enough that we can spin the test tubes and therefore kind of spin the flies around. And as they basically get exposed to more alcohol, we're flowing alcohol vapor through the tubes then as we do this. And then every couple of minutes, we spin the tubes and see how many of the flies, get knocked over and can't get back up. And for the first exposure, we do this for 40 minutes, and then we calculate the percent time to 50% sedation, what we call the ST50, very standard drug type behavior, right? The whatever 50% lethal dose, effective dose, whatever it is. So in this case, and for us, it's sedation time. How long does it take to get 50% of the flies in each tube sedated? We then let them recover and metabolize the alcohol, and that takes about 3 hours or so. And we repeat this process then. Same exact process, exposing to the alcohol vapor. And for a wild type fly or a control fly, they will take about 20, anywhere from 15 to 30 minutes longer, but it's usually around 20 minutes longer to reach that sedation point. So the difference between the second ST50 and the first ST50 will be about 20 minutes. And we call that the sedation tolerance time. So, which for shorthand, for this, we can just refer to as tolerance. And then because it's Drosophila, right, and it's got all these wonderful tools that we can use, that we can then manipulate different receptors or different transport vesicles and things of that nature to manipulate neuron activity using the tissue-specific tools that we have. It's called Gal4 UAS. And this allows us to manipulate tissues to manipulate the fly in a tissue-specific manner. So we can express transgenes in a tissue-specific manner and be very specific with how our manipulation is going. That's our basic setup, and our goal was then to just find new tolerance sites in the brain.
As a result of your research, what areas of the brain are involved in the tolerance effect of alcohol?
So the effect of my research, what we have found is a group of neurons that integrate sensory information into the circadian clock are necessary for tolerance. There are other brain regions that my lab and others had found. The learning and memory center is necessary. The kind of coordination center, I'll call it for the purpose of this. So we were trying to avoid those 2 areas because they've been kind of shown. The learning and memory center in particular is very important for tolerance. So we did a screen of over 100 different of these Gal4 lines, these tissue-specific lines, and we reduced the number of calcium channels in the neurons using an interfering RNA tool, RNAi. And we first demonstrated that it worked in the learning before we did this. And we found a couple of different lines that seemed promising. And so then we tested those, further. We used a tetanus toxin, which, as I'm sure many, in the audience, many physicians would know, right? Tetanus interferes with neurotransmission. That's why you need to get a tetanus shot. Because you don't want that to happen to you. So, but we're able to just express tetanus in like a couple of neurons, right? So that doesn't kill the fly or damage it, really. It just prevents those neurons from transmitting. And so we had a couple of different lines that we had found, 3 in particular, that we started looking for what the identity of these possible neurons could be in there. And so we started just doing basically knocking down different neurotransmitters in the brain in those lines. And we found that glutamate was necessary. which was exciting for us because surprisingly, glutamate hadn't been found to be involved in tolerance in flies before. Like we knew, obviously, right, in mammals, it's very, very involved in alcohol behaviors. But flies, I mean, they use glutamate in the brain, but not as much as we do. They in the brain they tend to use actually acetylcholine more, which is the other main excitatory neurotransmitter. You know, in mammals, we tend to use that more at the neuromuscular junctions. Right. That's why certain things, you know, like yeah, we don't go get too sidetracked. But so yeah, we found then that the specific grouping of neurons called the DN1A neurons they're glutamatergic, right? So they release glutamate, and they signal to the clock to help keep it in line with the environment. So they're clock neurons, but they aren't part of the master clock system, which in all animals, and I mean presumably many other things, but in animals the clock kind of has two parts. Fast oversimplification, and I'm not a circadian clock expert here, but you have the clock that actually maintains itself called the master clock, right? So this actually runs on about a 24-hour cycle. And then you have other neurons that are accessories to it. They'll have clock genes, but and so they have a up and down cycle of proteins, which is how the clock works. But it will drift if it's not attached to the master clock. And so the DN1As are some of these accessory clock neurons. And what they do is they seem to respond to both temperature and light. And they're part of a group called evening cells. So they activate and, you know, affect basically when the fly sleeps. And so if you get rid of these, the fly will sleep during the day at different times, and it'll sleep for longer because flies like to nap during the day, as well as at night. They tend to be most active at morning and evening. And so it responds to temperature and light. And so, you know, kind of our thought at the moment a little bit is perhaps they help keep the clock in line with like the seasons a little bit, right? Because it gets colder, it'll get darker. And so, yeah, we found that these glutamate signaling from these DN1A neurons is necessary for tolerance. If you knock it down, you get decreased tolerance development and increased naive tolerance as well. But that's a separate phenotype. That's their innate tolerance is completely different from this rapid tolerance, even though they tend to correlate a lot, but you can completely separate them as well. And then we were looking for what might they be signaling to. So we knocked down glutamate receptors in some actual clock neurons, or proper clock neurons, and we found that it repeated the same phenotype. So knocking down glutamate, LNDs, and the fifth LNV, and knocking it down gave us the same phenotype. So it reduced the tolerance development, and it increased the amount of time it took for them to sedate during their first exposure as well. So it seems like this system, this sensory integration into the clock system, is necessary for tolerance, which is interesting. Because like we've known for a long time, right, that alcohol affects your sleep, right? Drinking will cause you to get worse sleep. And likewise, we do know that sleep deprivation will change how you process alcohol, but we haven't really seen where it's to this degree of kind of connection where you need a functioning clock to just respond to the alcohol in a normal way. So that was kind of interesting. not 100% sure where to take that entirely, but
Yeah, and I find those two findings particularly exciting. One is the role of glutamate, as you mentioned, an
mhm,
excitatory neurotransmitter, being
mhm,
the key in terms of this type of tolerance. And the other
yeah,
one is the relationship with the clock. So my next question would go in terms of application in the future uh,
mhm.
to humans. So where do you think this connection can be clinically relevant between the glutamate and the circadian cycle and the clock?
I mean, that is a tricky one, because as I mentioned before, there is such a vast difference in kind of evolutionary spans. While there's a lot of similar functions, it can be a little difficult to get one-to-one connections, of like, oh, this brain region in the fly is equal to like this brain region. But I mean, if I'm having to hazard a guess, that let's say the hippocampus has similar functions in terms of clock cues and kind of integration. So that's a potential thing to look into. Um, you know, we do know that the mammalian clock does use glutamate as well. So there's a very obvious connection right there to look at. Um, as far as the specific, like a specific brain region, it would have to start off honestly pretty vague. I would, maybe consider the hippocampus. I mean, honestly, I'm not a mammalian brain expert yet. I'm working on that for my postdoc actually. I'm in mice now, but that is, that is still new to me. Most of my work previously has been in insects. So, uh, yeah, I mean, that would be my first guess as to where to look and possibly look for differences between people who perhaps do have higher tolerances, or obviously sticking first more preclinically in mice, say, right? The
Yeah.
hippocampus is a possible thing to look at.
Yeah, absolutely. And it's, I think the sometimes animal models they yield results, and some of them get lost over time,
Mhm,
and they don't translate into clinical medicine. But in this
of
particular
course.
case, the glutamate connection is something that definitely is advancing in the right direction.
Mhm.
The question is, for your group, what comes next after this finding?
I mean, for this particular aspect of it, there is a couple different things that could be going on. I mean, the project was a little bit wrapped up when I was last there about a year ago. So I don't know where they're taking it in regards to that. Uh, a little bit more at the moment at the chronic tolerance, which the lab also produced a paper in that. The one thing would be to pursue if we can locate what the upstream neurons are, so what is communicating with the DN1As. We were not able to determine what that was. So we tried looking at the clock, once again, staying within the clock. The communication molecule for that is something called pigment dispersing factor. Um, and so we knocked down the receptors for that in the DN1As, and we didn't see any effects. So either it's some other neurons upstream. As I mentioned, there are temperature sensitive neurons that connect to it. There's light sensitive neurons that connect to it. But, as I kind of alluded to earlier, this is alcohol. So it could just be the alcohol directly interacting with these cells. But that would be something to figure out. The easier path would be to continue the downstream pathway because we, have some good evidence there that these LNDs and the fifth LNDs are involved. So then we can start looking at what they connect to. The main problem you're going to run into pretty quickly is they connect to a lot of things.
Yeah, I bet.
Even the fly brain, as small as it is incredibly complicated.
Yeah.
So those are definite pathways forward for the clock aspect. Additionally, just within this paper, we found a line that contained additional glutamatergic tolerance neurons that we couldn't identify. They weren't part of the clock. So there are other glutamatergic sites of tolerance in the brain. So that's definitely something worth pursuing. Once again, just especially with how prominent glutamate is in the mammalian response and the human response to alcohol. And then we did find another line that we weren't able to identify in any way, shape, or form aside from what it wasn't. Just that has additional tolerance sites in the brain, so it'd be something for someone to pursue, an early grad student or something. Cause that's obviously going to be a lot trickier because we never identified what the signal was, so we don't know what it is. But those are the main paths forwards, right? So you can pursue the clock itself, which definitely has some promise, and has some complexity to it because the bright side, once again, a reason to Drosophila is useful is we've completed the connectome in the fly brain, so we know roughly what everything connects to. In a kind of generalized way, each fly is going to be a little bit different in the same way each human is going to be a little different in how the connections are, how many connections there are, what the strength is, and that sort of thing.
Mm-hmm.
But, that does allow for you to start pursuing, right? Okay, so we know like these LND clock neurons, okay, they connect to, 50 other neurons. Can we find lines that let us manipulate those? And that's a path forward, definitely, that would go down at least this path with the clock and that connection.
Yeah, and it's exciting to see that the different ramifications that come out of this, and obviously with the complexity of the brain, is going to take a long time. But the contribution that you guys already made is significant, and specifically in terms of circadian clock and glutamate. And I think this way we can wrap up our conversation by saying that that this is exciting. We are going to be following up on the research that you guys continue producing, and hopefully in the future we can meet and follow your progress. If you will.
Definitely.
Yeah, that's great. And first of all, I want to thank you for sharing your knowledge with us. And as I mentioned before, it's uncommon that we talk about animal models, but this one is really captivating, and I'm sure it's going to yield even better results. Thank you so much. And for our audience, you will find the link to the paper in the description, and don't forget to leave us your feedback. See you in the next episode.